The overall goal of this procedure is to quantify the spatiotemporal variations in the in vivo cell-free layer width.
This method can help to answer key questions in the field of microhemodynamics to better understand the role of cell-free layer in microcirculation.
The main advantage of this method is that the in vivo cell-free layer width can be quantified more consistently and conveniently than previous manual measurement techniques, which were very time-consuming.
Before beginning the cell-free layer width measurement, run the cell-free layer pre-MATLAB script file. Next, click Open File to select the video file for analysis and adjust the rotation slide to vertically align the walls of a single vessel using the zoom slide to adjust the zoom level as necessary. When the vessel is in the appropriate position, click Confirm Editing. Then click Set ROI to Crop to define the region of interest. The aligned image will be displayed in a pop-up window. Adjust the rectangular objective on the image as necessary, and double-click the objective to confirm the region of interest. Then click Extract Images to extract all of the edited video frames into consecutive bitmap images, which will be found in the folder with the same name as the selected video file. To measure the cell-free layer width, click Select Folder and click on the folder of images. The first image frame will appear in the Grayscale Image panel, along with a corresponding gray intensity histogram in the Image Histogram panel. Select